Yep, you guessed it, a pomegranate. If you watched our last video where we cracked the easiest way to remember mitosis and meiosis stages using a single fruit mnemonic, give yourself a high five. If not, well, go watch it, I'll wait. No, really, go. I'll be here, sipping my mitosis smoothie. So, in that video, we talked about the GR in pomegranate, remember? G for growth, which is mitosis, and R for reproduction, which is meiosis. Growth comes before reproduction, just like how you build your Lego base before going full castle mode. Now in today's video, we're diving deeper into what each stage of mitosis and meiosis actually does, and yes, we're still using the same tasty mnemonic, pomegranate. Let's start with mitosis, because just like your morning coffee, it comes first. P for prophase, P for prepare. This is where the chromosomes finally get serious and start preparing. They condense and become visible. Basically, the cell's been in pajamas all day and now it's putting on a suit. M for metaphase, M for middle. Chromosomes line up at the center like they're about to perform in a talent show. A for anaphase, A for apart. Now the chromosomes get pulled apart to opposite ends. Imagine two clingy best friends being forced to sit at different lunch tables. T for telophase, T for 2. The cell splits and forms two identical nuclei. It's a clean breakup, no drama, just two perfect clones. Alright, now let's talk about meiosis, and we're starting with meiosis I, where things get interesting. P for prophase I, P for paired. Homologous chromosomes pair up, and sometimes they even swap bits. It's not gossip, it's crossing over. Genetic variation, baby. M for metaphase I, M for matched. The matched chromosome pairs, called tetrads, line up at the center. It's like a blind date but way more organized. A for anaphase I, A for apart. But wait, we're not splitting sister chromatids yet. Nope, it's the homologous pairs that get separated. Sisters are still sticking together. Now, T for telophase I, T for 2. Now we've got two haploid cells, but the chromosomes are still doubled. It's like moving out of your parents' house but still wearing your old school uniform. Now on to meiosis II, which is basically mitosis showing up for an encore performance. P for prophase II, prepare again. There's no new DNA replication here, just more spindles and more drama. M for metaphase II, middle singles. Now it's the single chromatids lining up at the center, ready for their moment. A for anaphase II, apart chromatids. The sister chromatids finally split and move to opposite sides. Cue dramatic music. T for telophase II, total 4. And here's the grand finale, 4 unique haploid cells. Not twins, not triplets, just 4 genetically original individuals. Like 4 people who watched the same show but came out with completely different opinions. To sum it all up, PMAT for meiosis. Meiosis I, paired, matched, apart, 2. Meiosis II, prepare, middle, apart, total 4. And remember the key differences from mitosis. Crossing over happens in prophase I, that's your genetic variation. In anaphase I, it's homologous chromosomes that separate, not the sisters. And at the end of meiosis, you get 4 unique haploid cells, unlike mitosis, which just makes 2 identical ones. That's it for today's biolicious breakdown. If you found this helpful, confusingly funny, or just straight up weird in a good way, drop a like, subscribe, and share it with your study buddies. And as always, Stay curious, stay awesome, and I'll catch you in the next one. Peace.